Welcome to video 4 of the Search for a Better Health series. In this video, we're going to be looking at Pasteur and Koch and how their scientific discoveries have helped to contribute to our understanding of infectious diseases. And then we're going to look at, in particular, Pasteur's experiment, which we will perform in class as a first-hand investigation. So when we look at diseases in society, which formed most of the stimulus behind why people started to investigate microbiology and how that affected society is because we have things that we call epidemics, pandemics, and then plagues. When we talk about epidemics, we're talking about large scale, temporary increase in numbers of people being affected by an infectious diseases, and this may or may not be spread from human to human. Whereas when we look at a pandemic, we're looking at something that is infectious, that is affecting people across a large region, and it must spread from human to human. And then we look at things like the plague or plagues, which are similar to an epidemic, but aren't always associated with infectious diseases. So we can have a plague of rats or a plague of locusts. What Pasteur and Koch started from was the idea that of spontaneous generation. So this wasn't their theory, but this is what society believed was occurring, and people believed that living things could come into existence from non-living matter. And this was disproved in 1680 by Francesco Redi, and he showed that living matter must be present for other living things could appear. So this provided the background and the platform for Pasteur and Koch to work from. When we talk about Louis Pasteur did lots of things with infectious diseases and contributed greatly to our understanding. So he put forward the germ theory of disease, and he believed that most infectious diseases are caused by microorganisms or germs. Louis Pasteur examined samples of wine and identified that it was in fact microorganisms that caused the fermentation, which destroyed the theory of spontaneous generation. Pasteur was in fact responsible for developing the process of pasteurization, which is what we use today in milk and wines, and that's the process of heating products to 55 degrees for a few minutes, which can kill microbes. And then Pasteur was also his last little bit of contribution was his development of vaccines against anthrax, chicken cholera, and swine erysipelas. And this came about by inoculating a sample of animals with the anthrax virus, and this small sample size was in fact injected with a weakened form of the disease. And then when he gave the whole sample the full strength version of the disease, those that had been previously inoculated with the weakened strain were able to survive, with the other 25 unfortunately did not survive. So this was the basis that we use now for vaccinations. So I guess Pasteur's biggest contribution was the idea of disproving the theory of spontaneous generation. And he did this by using what we call S-shaped flasks. So he poured some broth into some flasks, and then he had these flasks, the necks of them bent, and these flasks allowed air to enter, but any spores and dust would be trapped in the curve of the neck. The process that he used once he had put the broth into the flask is that he makes sure they boiled it to kill any existing organisms. And we know that if we take that broth over a certain temperature, it ensures that we do kill any existing organisms in there. Then what Pasteur did is that he changed his two flasks so that both still had the same broth, but instead of both having the S-shaped flask. One of the necks of the flask was broken so that air could enter, and the other one still remained an S-shaped flask. The flask with the broken neck ensured that the air could enter and that dust and microorganisms could enter, whereas the S-shaped flask still ensured that dust and spores were trapped. And what he found was that the broth with the open neck became infected, whereas the broth with the S shape remained uninfected. So therefore, organisms must be carried in the air, which helped to disprove that theory of spontaneous generation. We then move on to Robert Koch, and he studied the cause of anthrax, and he in fact isolated rod-shaped cells and resting spores of anthrax in blood, and he demonstrated by isolating these that it was in fact the bacterium causing the disease as opposed to any other parts of the blood or the human body. Because of this, he described a criteria that must be met if a particular microorganism causes a particular disease, and this is what we refer to as Koch's postulates, which we'll look at on the next slide. And then his final, I guess his most recognized publicly discovery was the fact that he was able to isolate Mycobacterium tuberculosis, which is the bacterium that causes tuberculosis or TB. And in fact, he was awarded the Nobel Prize for developing tuberculin, which is the test for the disease. And tuberculosis was a pandemic which affected lots of people a few centuries ago. So when we look at Koch's postulates, these are 4 statements which allow us to understand whether the microorganism will in fact cause a disease. And if all 4 of these are met, then it does cause a disease. So the first one is the specific microorganism must be present in every host with the disease. Second, the specific microorganism must be isolated from the host and grown in a pure culture. A potential host, when inoculated with the microorganism, must develop the same symptoms as the original host. And the last is the specific microorganism must be able to be isolated from the second host and identified as the same species as originally cultured. These are his 4, and obviously he experimented on rats, and these are the 4 things which formed the basis of how we identified lots of microorganisms that cause diseases in society. Make sure you tune in for video 5 and make sure you do a bit of pre-reading for conducting Pasha's experiment in class.